Welcome everyone, and Lena, thank you very much for the nice introduction. My name is Ulrich Broeckel, and I'm the founder and CEO of RPOD Diagnostics. And today I would like to talk about how comprehensive pharmacogenetics accelerates personalized therapies and can reduce health and disparities for diverse populations. Health disparities and among ethnic groups in the U.S., it is well recognized that for racial and ethnic minorities in the U.S., health disparities really take on obviously many forms, including high rates of chronic disease, premature death, and that's compared to like other races, for example, like whites and Caucasians. We obviously recognize that the causes of health disparities are complex. There are socioeconomic factors which play a role, but if we're thinking about other factors, genetic variation is really now recognized that this also plays an important role in the disease development. And if we're thinking about then treating diseases, obviously the variation in the response to treatment, the drug response, exemplified by pharmacokinetics, as well as pharmacodynamics, can explain part of this variability among diverse populations. So we recognize in general, as we're thinking about pharmacogenetics, that the response of patients is not uniform. So we see clearly a variability that is really common knowledge in clinical practice. Not every drug works for every patient. And that relates not only to efficacy, but that also relates to the prevalence and the occurrence, for example, of side effects and the toxicity. So the goal of pharmacogenetics then is to really provide actionable data to provide a safe and more effective pharmacotherapy through the application of the genetic information, and obviously then guide also, if we're thinking about drug development, of identifying new drugs, guide drug discoveries and therapeutic decisions. When we're thinking about applications of pharmacogenetics, there are a number of different areas where this is currently applied at various levels. Obviously, in clinical research, as we try to better understand the variability of drug response, so to expand our knowledge base. There's also the component of obviously establishing clinical utility of pharmacogenetics and the application in that area. Another area where, and this is really growing, is the application of pharmacogenetics in the area of drug discovery. And there are, obviously plenty of examples with the advancement of precision medicine now that drugs now, the treatment of a drug or the selection of a drug gets particularly get linked to a genetic makeup of a patient. And the goals for that is that it obviously reduces the length time of and the complexity of the drug development process. And in drug discovery, we're thinking about going in clinical trials. Pharmacogenetics is applied in early as well as late stage clinical trials. And if we're thinking about clinical trials, we can think about the concept of preemptive or reactive pharmacogenetic testing, where we do the testing before a patient gets exposed to a drug or gets a drug prescribed. But we also can think about the other concept is the more reactive pharmacogenetic testing, where a patient receives a pharmacogenetic test after, for example, we observe toxicity or after we see a therapeutic failure or the occurrence of adverse events. In many cases, then obviously the results are not necessarily in a clinical research setting returned to patients. If we're thinking about, and ask the question, how frequent or what's the prevalence of where pharmacogenetics could be applied, I want to show you some results from a review paper published now about 5 years ago, when we're looking at the prevalence of how many drugs are currently on the market, where there are clear guidelines for pharmacogenetic testing. Overall, there are about 300 actionable genetic variants, which are linked to dosing guidelines and FDA-approved drugs. And currently, there are about 1,200, actually more than 1,200 drugs currently FDA-approved. And 7% of these drugs have clearly pharmacogenetic actionability results. Now, you can look obviously at the overall medication, but the probably more interesting way to look at this is as not every drug gets frequently prescribed and there's variation in prescribing patterns, you can look and ask the question, so of how many of the prescriptions filled in a year in the US, how many of these drugs and prescriptions which are filled have actionable pharmacogenes? And that's about 18%. So if we're thinking that about 4 billion prescriptions are filled in the US, 18% actually presents a substantial number of drugs and patients actually receiving these drugs. So that really speaks to the overall applicability and also the great potential utility of pharmacogenetic testing. When we're thinking now about genetic testing, and we started out with that, we wanted to expand this to diverse populations. One thing we need to recognize, and this is actually a phenomenon common to genetics research in general, and has been recognized just very recently, is that most of the genetic studies and studies which are performed to identify genes for diseases, but as well as for pharmacogenetics, these studies really have been performed only in Europeans or Caucasians. And ethnic minorities have really been historically underrepresented in clinical trials. So if you think about, for example, in 2011, African Americans in the US here comprise about 12% of the US population, but really only the participants, we only see about 5% of them participating in clinical trials. And on the right side, you can see kind of a breakup of the distribution, the ethnic distribution, what we normally see. And that causes and that directs us to a number of concepts where I would like to explain you and walk you through to explain why we think that very comprehensive pharmacogenetic testing, which is not only focused on the information what we obtain and the frequency of variants, which we mainly see in Caucasians, is really critical, and we really need to expand in this area. So one of the things which has been recognized quite early on now is that the allele frequency or the variance of star alleles differ between ethnic groups quite significantly. And so just to briefly explain, star alleles in genes, in pharmacogenes, we analyze many different DNA variants. These DNA variants are not necessarily independent of each other. They travel together. They are linked to each other. And so you can identify subgroups. You can identify subgroups of DNA variants and group them then together in what we call haplotypes or star alleles. So as we're looking, for example, at a number of very commonly genotyped or tested, analyzed genes relevant to pharmacogenetics, for example, CYP2C19, CYP2D6, or CYP2C9. You can see in the different colors the frequency of these different star alleles in different ethnic groups. So, for example, if you look at CYP2D6, you see that some of these star alleles are pretty frequent in one population, for example, star 4, and they're not that frequent in other ethnic groups. And that has actually then implications for pharmacogenetic testing. For example, if we look at CYP2C9, to give you an example, star 8 and star 11 are genotyped in panels and really have been shown to improve the warfarin dose prediction, the required dose for anticoagulation, particularly in African Americans. So to give you an example of the importance of testing of rare alleles, again, thinking about, for example, warfarin, BCOR-C1 has been linked to the response to warfarin and the required dose to reach a certain level of anticoagulation in patients who require that. There's a certain variant which provides great contribution to the variability in dose among Europeans. And that's obviously has a higher contribution in Europeans compared to non-Europeans, and that is largely explained by the difference in the frequency of this variant, this particular variant. Similarly, for CYP2C9, the star 5 haplotypes, star 6, 8, and 11 are identified in African Americans, but essentially are absent in non-African populations. In addition to that, there was a genome-wide association study published which identified a novel variant for a particular SNP and the warfarin clearance in African Americans, but not in European or Asians. So what that really shows is that these allele frequencies and also the presence of certain variants, which are unique to ethnic groups, are of significant clinical importance. So I want to show you some example, and I want to dive a little bit into a complex gene where we again can show that the difference in allele frequencies and the frequency in haplotypes, the complexity, has really important implications for the selection of drugs in pharmacogenetics. So I want to talk about one of the most commonly tested genes for pharmacogenetics, that is a CYP2D6. It's involved in the metabolism of about 25% of all clinically used drugs. It is a highly polymorphic gene with right now over 114 distinct groups of variants, 113 star alleles, haplotypes. What complicates that, and you can see this on the right side, on the top example, the gene CYP2D6 lives right next to a pseudo gene CYP2D7, which is also highly homologous. So these genes are very similar in their DNA sequence, and CYP2D7 is a pseudo gene. Now, pseudo genes do not get transcribed. They basically are just part of the genome and presumably do not have any function. What adds to the complexity now of analyzing 2D6 is that, as you can see on the right side, there are complex structural variations in this gene. For example, duplications, we can have deletions, and what makes it even more complicated for the analysis that CYP2D6 and 2D7 can merge together. Parts of the gene can merge together and form so-called hybrid alleles. So that really provides great challenges for the DNA analysis as we try to genotype this particularly clinically important gene. So I want to talk a little bit about the efforts of analyzing not only the common variants for 2D6, but also the rare variants. And I want to draw attention to an initiative which is really based at the CDC. It's called the Genetic Testing Reference Material Coordination Program, a long name, and the abbreviation is GetRM. And GetRM's goal is really to provide a reference material, DNA samples, cell lines for interesting groups, but also characterize these samples with all commonly available clinical testing platforms, and now in particular here for pharmacogenetics. So at RPRD, we were one of the partners and participate in this reference DNA sample characterization projects, and we provided the data using Thermo Fisher's PharmacoScan platform and analyzed about 179 DNA samples. And what we could show here, and the data is publicly available, and we would be happy to also share the link for anybody who's interested. We actually found this, using this assay, we found rare alleles, not only in CYP2D6, which have not been described in other genotyping platforms or were not detected in other genotyping platforms because some other platforms are not testing really these rare variants. This platform also allows for the detection of duplication and deletions without the need for any additional secondary methodology like add-on tests, for example, to detect duplications. And the platform also allows for the analysis of these, what I described to you, complex structural rearrangements between CYP2D6, 2D7. And we're very proud that the CDC recognized this project with the Shepard Award for Laboratory Science just this year. And below you can see a link to the publication if you're really interested in that. And the DNA samples are available for purchase, if any labs are interested in obtaining them, to use them as a reference material. And the clinical and the genotyping results in all these platforms are also available for download. So I want to show you one and talk about one example where we participated by genotyping a sample, And this was a case report just published in Genetics of Medicine, together with our collaborators at St. Jude's Children's Research Hospital. So this was a 18-year-old male African-American who was enrolled in the PGen for Kids preemptive pharmacogenetic clinical trial, which is a clinical trial genotyping all patients who are seen at St. Jude's Children's Research Hospital for clinical care, in particular for pediatric cancer. So we genotyped this particular patient with the DMET platform, and that DMET platform, which is the previous platform for PharmacoScan, and we detected for 2D6 a genotype or a diplotype of either star 4, star 40, which would make this patient individual a poor metabolizer, meaning that this enzyme has very little activity, or we detected, or we could not conclusively determine it could also have been a star 10 and a star 40, which would have translated an immediate metabolizer. Now, the difference between a poor metabolizer and a medium metabolizer is clinically highly relevant because obviously this affects the dosing of many potential drugs. And the question for this particular patient was related to the use of opioid analgesics, which are metabolized through CYP2D6, and obviously the correct dosing for this class of drugs is really important to prevent or reduce adverse events. The sample then was tested because we couldn't, that with this old platform, could not detect a unique metabolizer status. Was sent out to a different lab, and this lab tested, and the report came back as a star 4, star 17, which would have translated this to a intermediate metabolizer, and that was the diagnosis at that time. Now we started, because this was the previous platform, we now started to genotype this sample at our PID using the latest Pharmacoscan assay. And we actually, in fact, confirmed that this patient was a star 4, star 40 poor metabolizer. And I told you the CYP2D6 is a highly complex gene. And so just to dive into, we started to dissect, the differences in the different genotyping platforms. So the star 40 haplotype really differs from the star 17 haplotype by the presence of an in-frame insertion variant, a particular insertion variant. And if this variant is not probed, then what most of the testing platforms do will default to the call of a star 17. And as we mentioned, in the previous slide, these differences are clinically significant and obviously variant, important and relevant, because the star 17 has an activity score of 0.5, while the star 40 has an activity score of 0, so there is no enzyme activity. So the other important piece of information, which is to keep in mind, is the star 40 in CYP2D6 is really commonly observed in African Americans in about a frequency of 0.17%. So while this is not really very frequent, this variant, nevertheless, it is clinically highly significant and relevant to dissect even at rare variants particularly if you're analyzing non-Caucasian populations. So most institutions implement CYP2D6 genotyping, and they might not include this variant and therefore have the chance or the risk of potentially assigning a inaccurate phenotype. And with that example, I think we wanted to show that comprehensive coverage, not only for common variants and common SNPs, is really important, that we really very comprehensively analyze all pharmacogenetically potentially important variants, especially if you want to apply pharmacogenetic testing to diverse populations. So allele frequencies obviously now vary between different ethnic groups, and again, as I said, the subject can be at risk for misclassification if a particular rare allele is not included in a test. And I just wanted to emphasize again some of the FDA's recommendation that the selection of genetic markers needs to be appropriate for the population being studied and should include also rare functional alleles in the analysis, in addition to translating the genotypes then into the relevant drug metabolizing phenotypes. So this really emphasizes and underscores our approach to not only test common variants, not only test variants which have been identified in the populations which are frequently studied, but really include also rare variants and make sure that you're testing in a broad-based and set of markers which are relevant to a broad set of populations. I just want to highlight a few things about this array. The array itself includes about 2,000 relevant pharmacogenes in about 150 genes covering, as I said, diverse populations. It is obviously fully configurable. It is a single assay offering both copy number as well as HLA analysis. And depending on the genotypes, you can translate this. There's software available which translate this into star alleles. And if you're interested in clinical research, then there are partnerships with, for example, Coriell and Life Science and other vendors who can guide and use this information for medication management reports. So it has broad applications because it really includes pretty much all pharmacogenes you can think of right now, all the ADME status genes, all the genes for GKB level 1 and 2. So it has broad applications across many therapeutic areas, for example, in cardiology, psychiatry, neurology, pain management, and also perioperative markers. So it's broadly based, not only targeted to a particular disease area. And as I said, it's very comprehensive. The coverage includes also all the markers which have been recently approved and recommended by the Centers of Medicare and Medicaid Services as a psychiatry panel. It also includes the Dutch PGX passport and includes, if you're thinking of different platforms, the catalog of the open array TACMAT panels. In terms of application, we see a great use and the ability to use this platform for preemptive screening in targeted pharmacogenetic content as a high throughput and low cost solution. So here's just a little bit more now extension on pharmacofocus. Here the content, about 88% of these variants reference to CPIC guidelines with high level of confidence. It includes copy number calling for high value genes, for example, COIP duty 6, where I told you about the copy number variations we observed, but also COIP 2A6, GSTM1, 2, UGT2B17, and ULT 1A1. It also allows for HLA genotyping of 11 loci, and again includes the coverage for markers found in the open array panel. What's particularly, I showed you the example for the difficult genotype 2D6 gene applies to other genes, and again it translates this to about 880 star alleles across the genes. So if star alleles are available for a particular gene, the star allele nomenclature translates that as well, and the platform itself is available for 96 and also in a 384 well format for high throughput analysis. We did a comparison between the Pharmacofocus platform and the PharmacoScan platform, and just to show you here some examples, across the board we see over 99.8% concordance between these calls between the different platforms. Same translates to, this is on the genotyping level, and if you look at the star allele level for the genes where we have star alleles, we see a very high concordance between the two genotyping platforms, showing the great comparability between those two tests. The other thing what we at RPID offer now, together with genotyping these platforms, is we have over 18, close to 20 years experience in translational genomic research. We provide in-depth analysis and custom interpretations and end-to-end consulting particularly for pharmaceutical customers, customers in the drug development process, where we help and consult on preclinical studies, study design for early phase clinical trials, late phase trials, as well as post-marketing. We contribute to answer some of these questions, like, the interpretation of variation in pharmacokinetics, the genetic analysis for the individual variability in drug response, if you observe this in a clinical trial, the genetic analysis for the lack of efficacy, for example, or the occurrence of adverse events or toxicity, what you see. The other approach, what you can look is, obviously, you can try to enrich your study for responders in late phase clinical trials using genetic information. I think this is an approach which in the future actually will gain quite significant interest, or already has some interest, but will really expand. We are a CLIA certified pharmacogenetic diagnostic testing laboratory, and our team offers the full spectrum of pharmacogenetic services that includes pharmacogenetics high throughput scan and custom panel analysis. As I mentioned, PharmacoScan, PharmacoFocus are platforms we offer in addition to next generation sequencing, as well as targeted sequencing analysis using, for example, the AmpliSeq platform. We provide in-depth data analysis and custom clinical interpretations. And as I mentioned already, we're also providing consulting services for the pharmaceutical industry related to clinical trials and planned research projects. We're very innovation-driven, and we provide these solutions in a world-class scientific research environment. We've been really bringing preemptive pharmacogenetics into clinical use. The value of preemptive testing is that if patients have genetic information, pharmacogenetic information is already at hand, you can use this information right when you want to prescribe it. If a physician wants to prescribe a drug, doesn't have to order a test and then wait until the results coming back. Having preemptive pharmacogenetic information available before you have to make a prescribing decision has significant clinical workflow implications. So we've been piloting this with a number of our customers and collaborators now for many years, and we're demonstrating the value of this. And we really offer high quality pharmacogenetic testing in this environment by many healthcare systems here in the US rely on our services. So taken together, I hope I could show you some of the offerings, what RPID brings to the scientific and clinical community. The key points, what I wanted to emphasize again is that obviously, and I hope I could show you that the lack of diversity in genetic research and clinical trials may explain part of the ethnic health disparities we have in the US. It's a well-known fact that allele frequencies differ among different ethnic groups, and we really need to keep that in mind, not only for clinical testing, but also for clinical research. And as we generate more pharmacogenetic information, we really need to keep that in mind and recruit minority populations in our research programs. The discovery of many ethnic-specific alleles shows the importance of comprehensive allele coverage in pharmacogenetic testing, especially if you're testing diverse populations. And the PharmacoScan and the PharmacoFocus platform provides comprehensive allele coverage, which is an important consideration when you're investigating and testing diverse populations here in the US and abroad. PharmacoFocus assay demonstrate a high level of concordance between the PharmacoScan assay. And RPD, as a team, we have nearly 20 years of experience in pioneering translational genomics research. And so we continue to provide in-depth genetic analysis and interpretation to guide clinical trial design and improve the discovery process. So with that, thank you very much for your attention, and we will have some questions.